morning, everyone, and thank you for being here. My name is Isabel, and I am a master's student in the Bruno Lab at the University of North Carolina at Chapel Hill. I am very excited today because I flew all the way from the Galapagos, where I am currently based for the spring, to briefly present the project I am conducting for my thesis down there in San Cristobal Island. The project explores trophic interactions in the shallow subtidal rocky reefs and how they are mediated by ecological parameters in this upwelling system. In this picture, we have one of the most common reef fish species, an adult Mexican hogfish preying on one of its favorite prey items, a small pencil urchin. The Galapagos Islands are highly influenced by a complex oceanographic setting. We have major warm and cold currents that hit the archipelago and create an enormous spatio-temporal thermal gradient across islands. The convergence of currents also result in seasonality and in contrasting productivity regimes shaped by upwelling. Thank you, Colleen, for this graph. This work is being done on Cerro Mundo Bay, southwest of San Cristobal Island. And this down here is a 2-year temperature graph of our study site from July 2019 to January 2022. The gray lines are temperature every 30 minutes and the red lines are daily average. As you can see, there are two marked seasons. We have a cold season associated with high upwelling that peaks around September, and we have a warm season associated with low upwelling that peaks around March. If you see on the y-axis for this site, for this period, we see a 13 degrees Celsius difference. Moreover, it is known that trophic interactions can be mediated either by top-down or bottom-up forces, and that temperature is a major driver at multiple scales. So on the A side, this is a conceptual model showing the hypothesized effects of temperature on the trophic interactions in this tropical upwelling region. So on the A side, temperature affects metabolic rates of ectothermic species, which results in changes in grazing and per capita consumption. And on the B side, the role of nutrients varies as well. Nutrients enhance primary productivity via algal growth, exerting bottom-up control. On the other hand, a high nutrient concentration can also reduce herbivory by decreasing algal palatability or due to herbivores altering their feeding rates in accordance with nutrient concentrations in algal tissue. So for this project, I want to focus on the B side of this model with the purpose of studying the interactive effects and the importance of herbivory, temperature, and nutrient availability on standing macroalgal biomass as a proxy for primary productivity. So inspired by John Whitman's work and his pizzas in 2007, we have our concrete pizzas, and specifically we have 4 treatments. The first one is an open cage, open to all herbivores. The second one is a herbivore exclusion cage with sides and tops to exclude all herbivores from grazing on the concrete substrate inside. We have a third treatment, which is a herbivore inclusion cage, where we put 2 pencil urchins inside. In this way, we hold the identity and the density of our herbivore constant. And do not worry, we didn't cage John inside this treatment, but yeah, there he is. And we have a procedural control with partial sites. Additionally, half of these treatments receive nutrient diffusers, so a diffuser with a fertilizer. We have 4 treatments with 2 nutrient categories, ambient and elevated, and we have 8 replicates each. which leaves us with 64 cages deployed on the rocky reef at around 9 meters deep. With these treatments, we can manipulate the variables herbivore presence and nutrient level, and we take advantage of the natural thermal seasonality of the archipelago to control the variable, well, to use the variable that we can't control, which is temperature. So another set of data that we are collecting on this site, of course, temperature. We have loggers deployed to be able to characterize the thermal profile of the reef yearly long. We are also doing visual and GoPro censuses. So by doing transects and by deploying GoPro cameras, we can characterize the fish community throughout seasons, and also how this might change in the event of a temperature perturbation. And really important, we can also determine which species are interacting with our treatments and the type of interactions they are having. And lastly, we are also collecting water samples and algal tissue, and in this way we can test the effectiveness of our nutrient diffusers, and we can test for concentrations of nitrogen and phosphorus constituents. So the trials run for about 4 to 6 weeks. We run them multiple times through the year because we want to capture the effects of a wide range of temperature of a thermal setting. And additionally, we do weekly replacements of our diffusers. At the end of each trial, we scrape and vacuum all the algae that has accumulated on each experimental unit. We use an underwater vacuum. This is a short clip of the vacuuming process that we do at the end of each trial. And experimental ecology is super fun, especially if you get to hang out with these Mexican hogfish friends. However, sometimes these science buddies bite your fingers while you're diving, but that's fine. Anyways, the accumulation of macroalgal biomass is a response variable, so we bring this algae back to the lab, we dry it, and we burn it. So these are some descriptive and preliminary results. These are some pictures of some fauna that our GoPros and us have recorded on site. With GoPros, we can record some species that we definitely do not see while diving. So a marble ray, we have this seahorse, which in theory you only see it in Isabela in the west. This is on the east, and I've only seen seahorses on this site. We have bullhead sharks, we have green sea turtles, these bullseye pufferfish grazing, and some unidentified shark species as well. So far we have 45 species observed on the vicinity of our experiment, and 10 species grazing on the pizzas. So this is a sequence of pictures of how algal growth looks in the absence, top row, and presence, bottom row, of herbivores. So on the top row, we see this flourishing community of macroalgae. We have some Dictyota, some Ulva, and some red filamentous algae as well. And on the bottom, we see how urchins are constantly grazing on the concrete. After deployment, we could tell how quickly algae colonized the concrete, and we observed an indistinguishable algae community from the adjacent natural substrate. To me, this picture specifically, it's kind of a border effect. The yellow arrow points to the mesh, so to the wall of the pizza. This is a herbivore inclusion cages, so to the right, herbivores, urchins are heavily grazing the substrate, and then right on the border we see this ulva, which grows on the concrete. and also we have the main macro grazers frequently interacting and grazing on our pizzas. So these are results from pilot trials that we did a few years ago. These are in the process of getting published, and due to COVID-19, these trials abruptly ended. But since the end of the summer last year, the Galapagos National Park granted us permit to do field work again in the marine reserve. So we restarted the experiment with some modifications. So on this graph, we have on the y-axis dry algae weight. On the x-axis, we have nutrient categories, and the colored bars are the treatments. So far, excluding herbivores has a clear effect on the standing macroalgal biomass. And macro herbivores really have a strong effect on the benthic algal community in the Galapagos. Dry algal weight, the gray bars, was very similar for open pizzas across seasons. So this suggests that at least for this period, temperature did not have a strong effect on grazing intensity. This was opposite to what we were expecting, which was that on warmer conditions to see a stronger predatory control due to an increased metabolic activity of macro herbivores. However, this effect of temperature can be further elucidated by running other trials during thermal extremes, so during the peaks of the warm and the cold season, to see what is happening there. Regarding nutrient concentration, the highest increase of dry algal weight was recorded for for open pizzas on the cool season when nutrients were added. Even though this was a non-significant difference when for open pizzas on ambient nutrient levels, this could hint that herbivory was reduced through a high nutrient load on algal tissue. For the warm season, the dry algae weight on herbivore exclusion cages increased twofold when nutrients were added. This idea is consistent with the idea that on warmer conditions, nutrients are limited due to the weakness of whaling. Finally, I want to say that this is a collaborative project and effort with a lot